Us. Today we are joined by Dr. Colleen McClung. Dr. McClung is a professor of psychiatry and clinical and translational science at the University of Pittsburgh School of Medicine. The research she will be sharing today explores how circadian rhythms influence psychiatric and substance use disorders with the goal of developing better treatments for people who struggle with those. She has published over 100 papers, received numerous awards for her groundbreaking work, and she currently directs the NIDA-funded Center for Adolescent Reward, Rhythms, and Sleep. She also does amazing work with numerous organizations outside of that. So I will go ahead and send it over to you. She has a great presentation for us with a lot of really good information. So I will let you take it from here.
All right. Thank you very much. Let me try to share here. All right. You guys see this?
Yes, we
Okay,
do.
great. All right. So thanks for having me. So today I'm going to share a little bit about circadian rhythms and bipolar disorder, including some of our work and some sort of general information about what's going on in this really exciting field. So, just to begin, circadian rhythms are our body's 24-hour clock. Basically, everything that goes on in the brain and the body revolves around this sort of 24-hour day. So, things like your coordination time, your cardiovascular activity, blood pressure, bowel movements, sleep, of course, is all sort of regulated by the circadian clock. And the way that this clock is regulated is light input that comes in through the eyes can impact a part of the brain called the suprachiasmatic nucleus, or the SCN, which is located at the base of the hypothalamus. And the SCN kind of has its own internal synchronization mechanism. So even in the absence of light, it will run on about a 24-hour schedule, but light will just synchronize it each day. And it's really the most important synchronizer that we have, although there's some others as well. And then that clock in the brain can send projections out throughout the brain, throughout the body to control physiology and behavior. And we really are just a coordinated organism of clocks. So pretty much every cell in the body, in the brain, has its own internal molecular clock, and they're really all functioning in sync with one another. Ideally, as long as everything is synchronized properly. As I mentioned, light is really the most important driver, but things like the time of day that you eat, time of day that you exercise, things like that can also help to kind of entrain the body's rhythms and make them all in sync with each other. And of course, things that we do in the environment can put us out of sync. And so things like shift work, if we're working night shifts as opposed to day shifts where people are diurnal, and so they really we're supposed to be awake during the day and asleep during the night, but of course shift work can disrupt that. Things like jet lag. Obviously, artificial light at night can disrupt circadian rhythms. And teenagers are especially prone to this, where they have a really wacky schedule sometimes where they're waking up really early in the morning to go to school, and then they're staying up late at night, and then they're sleeping in on the weekends. So this can really cause a lot of disruption to the circadian clock. And so there's, ideally you like your clock to be kind of in line with the environment and with the time of day you wake up and the time of day you go to sleep, to match the sunrise and then going to sleep in the evening. But there's a lot of things that happen in life that really just can cause a lot of desynchronization to this clock. At the molecular level, these clocks in the cells are controlled by a transcriptional translational network. So there's clock genes. The one that is most important today that I'll talk about a little bit with some of our studies is called the clock gene. and it binds to a protein called BMAL1. And these can regulate the expression of a bunch of different genes in a rhythmic way, including the period and the cryptochrome genes, which can then, once they're made into proteins, can feed back and regulate the expression of clock and BMAL1. So this really sets up this loop of gene expression that's happening in every single cell in the body, with very few exceptions, over a 24-hour period. And again, all of this leads to all the various rhythms and things that are important in human physiology. So what about circadian rhythms in bipolar disorder? So we know that in bipolar disorder that it's very common to see rhythms when they're measured by something like an ActiWatch, like something like this, actigraphy, or when we do questionnaires with people about their circadian rhythms. It's very common to see that their rhythms are abnormal, that people with bipolar disorder often have rhythms that are shifted a bit, or they're very irregular, or they have what's called a late chronotype, where you want to kind of be a night owl, or you want to stay up late and wake up late. And this is very common with bipolar disorder. So all of these things are very strongly associated with the disorder. We also know that these things that can disrupt your clock, like shift work, jet lag, also seasonal changes, can trigger or exacerbate mood episodes. This is very, very common in terms of precipitating manic episodes or depressive episodes. We also know from genetic studies that polymorphisms in those circadian genes that I talked about are associated with bipolar disorder. So genetic studies have shown that this might be a system that's involved. And we also know that targeting circadian rhythms can have therapeutic effects in some individuals with bipolar disorder. So things like bright light therapy, interpersonal social rhythm therapy, where you put people on a very strict sleep wake and social schedule. These things can have some therapeutic benefits. There's also been some really interesting studies that have been published lately that have used, again, like things like actigraphy, these ActiWatches or Fitbits or Apple Watches, to try to predict when somebody's going into an episode. And this was a recent study that came out from a group in Korea, where they found that if they had a year's worth of data on people's activity rhythms, then they could actually predict a few days beforehand when somebody was going to have a manic episode or when they were going to have a depressive episode. And it was actually based on their circadian phase. And so which means that sort of the timing of their rhythms. So if their rhythms were running a little bit forward, so things were just shifted a little bit more towards the morning, they were more likely to have a manic episode. And if they were shifted more towards the evening, they were more likely to have a depressive episode. And so this is some really interesting work that's coming out that might be able to help physicians when they're trying to predict and intervene when someone's about to have an episode. And this goes along with some gene expression data that a similar group had published, which showed that when you measure expression of those circadian genes in cheek swabs, so they actually take cells from the cheek, and they do gene expression on it. If the rhythms were running a little bit fast, running a little forward, then the people were more likely to be in mania. Or if they were running a little slow, they were more likely to be in depression. This also kind of goes along with some anecdotal reports of when people fly to the East, like traveling from the US to Europe, they're more likely to have a manic episode, or if they're flying west, they're more likely to have a depressive episode because you're shifting rhythms forward or shifting rhythms backwards. So these are some really interesting studies that are starting to come out that have taught us a little bit about, what can predict using circadian measures, whether a person is going to have an episode and what kind of episode they're going to have. As I mentioned, there's a lot of sort of chronotherapies or circadian related therapies that can help people with either bipolar disorder or depression. Things like bright light therapy. You have to be a little bit careful with bright light therapy with bipolar disorder, as it can sometimes switch people into mania if it's given early in the morning. But it can also be beneficial for certain people. Total sleep deprivation is actually a really commonly used short-term antidepressant for people with major depression. It doesn't last very long, though. It only lasts till someone goes to sleep again. Social rhythm therapy, I mentioned, it's where people are put on a very strict sort of wake up and go to sleep schedule. And then medications also, like melatonin, which doesn't work all that well, honestly, as a therapy, but some people use it kind of as an adjunct. Um, Lithium and valproic acid, I'll get back to this kind of at the end of the talk, are known to have some effects on circadian rhythms, as well as SSRIs can impact circadian rhythms. And so a lot of these treatments that have been commonly used for mood disorders actually influence the circadian clock, and this might be a way that they're having therapeutic effects. Here's an example of social rhythm therapy. So you can see on the left-hand side, these are activity measures of somebody with bipolar disorder. The black is when they're active, and the white is when they're asleep. You can see that this particular person had very irregular rhythms, where their rhythms are just kind of all over the place. And when you measure their mood, excuse me, the negative is depressed and the positive is manic. And you can see also their mood state is sort of all over the place. But then when they're put on a very strict sleep wake schedule, their mood also really stabilizes. So again, really reinforcing this idea that if you can get your circadian rhythms in line and keep them very stable, this can be therapeutic. Um, Another thing that's really interesting about bipolar disorder is the seasonal component. So it's very common that people will have seasonal changes associated with episodes. And this is from a review which shows hospital admission rates. And you can see that in the spring and in the summer, there's usually more admission rates for mania, whereas in the fall and in the winter, you get more depressive episodes. And this is quite common for people with bipolar disorder. So I'm going to talk about a little bit of data that's from our lab. So this is actually unpublished data that were a very preliminary study that we have been working on. And here we're using human brain samples. So these are brains that are from people who died and they donated their brain to science. We have a really fantastic brain bank here at the University of Pittsburgh. And we decided to look at the prefrontal cortex, which is a really important region of the brain that's involved in the regulation of executive function, sort of top-down control of impulsivity, reward seeking, other processes like that. It's also involved in working memory and cognitive processing. And we know that disruptions to the prefrontal cortex are associated with cognitive dysfunction, mood changes, and impulsive behavior. And the regions of the brain that we decided within the prefrontal cortex are the dorsolateral prefrontal cortex, which is labeled here, or the anterior cingulate cortex, which is labeled here. So our approach to studying seasonal patterns in the brain is if we knew the person's date of death, we were able to basically put the brain samples along a 365-day axis based on the day that they died. And we had about 249 subjects, and we took those different brain regions. We took those samples and then we sequenced them for gene expression changes, or just their overall pattern of gene expression. And then we basically fit that along a curve according to the time of year. So In this case, this would be a gene that was peaking in about the summer months and coming down, getting into winter. And so this is just sort of another example of that. So you can see we have individual subjects that we put around a calendar, basically. And then you can see if a gene has a change in expression over the course of a year by measuring the entire population of people like this. So this is again a gene. Here would be an example of a gene that was had the peak in the winter and spring and kind of a trough in the summer. So basically, we sequenced people who had who were either control subjects, so no psychiatric disorders, subjects with major depression, subjects with schizophrenia, or subjects with bipolar disorder. And we saw how many genes just tend to have this kind of seasonal pattern. And we saw that in the ACC, there were several different genes. Depending on how strict you make your significance cutoff, you get a different number of genes. And we had several in the ACC and in the DLPFC. But what was really striking is that with the major depression group, um, that we saw a lot of genes that had a seasonal pattern in the ACC, and with the bipolar disorder group, we had a lot that had seasonal patterns in the DLPFC. Now, these two regions are a little bit different from each other. The ACC is kind of a deeper region. It's more involved in sort of mood regulation, whereas the DLPFC is sort of more of a higher level, more involved in cognitive control, impulse control, things like that. So it was interesting that we saw a difference between major depression and bipolar disorder in terms of having a lot of seasonally rhythmic genes in these two different areas. And we actually didn't see a whole lot of seasonally rhythmic genes in the controls. And so this was also really interesting that they tend to have a lot more seasonally rhythmic genes in mood disorder than in people who don't have a psychiatric disorder, and also more than people with schizophrenia. And schizophrenia is not typically much of a seasonally impacted disease. And so, so this was really interesting to us that that the mood disorders have just seem to have more rhythmicity in their gene expression than the controls or schizophrenia subjects. When we looked at when these genes are peaking, we found that in the bipolar disorder group, it definitely seems to be mostly in the summer. And so, and this is again when you were normally going to have more of a presentation of mania is in the summertime. And so this kind of goes along with this, that maybe these expression changes that we're seeing in the DLPFC, excuse me, are involved in the manifestation of mania in the summertime. With the major depression, we tended to see more of that peak of genes in the fall and winter, and that's more of when we see sometimes seasonal patterns with major depression and of course with seasonal affective disorder. And so those genes might be more involved in the depression related changes that happen in those subjects. We also found a smaller peak in the spring associated with major depression, but it was much smaller compared to the massive peaks that we saw in for the summer in bipolar disorder, and even less so than the winter for major depression. When we looked at what these genes are in bipolar disorder, we found that the vast majority of them are involved in mitochondrial function. And so mitochondria are the powerhouse of the cell. They're these little, organelles that are in the cell that give the cell its energy. And so it was really interesting that when we see these peaks in season in gene expression, particularly in the summer, when it's associated with mania, we see this very strong change in genes related to mitochondrial function. So this suggests that there's really these changes in energy that are happening that are associated maybe with that seasonal change to mania. And we know that there's a very intricate relationship between the circadian clock and that goes on in the nucleus. So, as I mentioned, this transcriptional translational feedback loop between the clock gene and the purr and the cry genes, that all happens in the nucleus of the cell. But they interact with the mitochondria of the cell through these sorts of um, pathways that are affecting reactive oxygen species and energy production. So there's a very intricate relationship between the circadian clock and the mitochondria. So we're really interested in teasing this apart a little bit more and figuring out how these seasonal changes are happening and how they might lead to mania. Okay, and so now I'm going to switch gears a little bit and get into our mouse work. So in addition to our work in human postmortem brain, we also work on mice. And the reason we work on mice is because, we're able to model some of the aspects of bipolar disorder using mice, although not completely, obviously. We can just measure sort of some facets of it. And of course, we can do a lot more invasive studies in mice than we can in humans. And particularly, we're limited in our human studies to people who've already died. And so, this is the mouse studies and the human subject studies are really complementary in a lot of what we can do. So, the mouse that we work with a lot is a mouse that has a mutation in the clock gene. And so, as I mentioned, this is one of the central regulators of circadian rhythms. These mice were discovered by Joe Takahashi and Fred Turek and Martha Vitaturna many years ago, back in the 1990s. They have a mutation in this particular gene, and it leads them to have abnormal circadian rhythms. So they will go arrhythmic if they are in constant conditions, but they also have this kind of longer circadian period in their activity. And when we tested these mice in a variety of assays, we found that they really are very similar to people with bipolar disorder, specifically in the manic state. So they're very hyperactive. They sleep less than wild type mice. They have less depression like behavior. They're more impulsive. They have more novelty seeking, more risk taking in a number of behavioral models. And they also are more sensitive to the rewarding effects of everything. So, cocaine, alcohol, sugar, brain stimulation, anything you do that's rewarding, these mice love it. They go crazy for it. They also gain more weight because they really like to eat. They like to eat a lot of food. Um, and if we give them lithium or valproic acid, we're able to reverse these behavioral phenotypes. So this makes the clock mutant mice a really powerful tool for which we can use to help discover some of the cellular and molecular and circuit level changes that are responsible for bipolar mania. So because of their phenotypes, we decided to focus a lot on this area of the brain called the ventral tegmental area. It's definitely a reward and mood related part of the brain. This is also where a lot of dopamine neurons are located. So dopamine is a neurotransmitter that's really important in reward. It's really important in activity, in a lot of the different motivation, a lot of the different things that are involved with mania. And so we really decided to focus on this part of the brain in these mice. We've done additional work in the prefrontal cortex as well, but I don't have time to tell you about everything today. So I'm going to focus on the dopamine cells. So we did electrophysiology in these mice, which means we just recorded the activity of their dopamine neurons. And we found that they indeed have increased dopamine cell activity. And when we gave them lithium, we actually see a reduction in this. And so basically, they have increased dopamine cell activity, and yeah, lithium is therapeutic in the sense that it's reversing that phenotype. We didn't know because clock is expressed throughout the body, throughout the brain, if this was due to a loss of clock in in these dopamine neurons or if it was due to a loss of clock somewhere else. So we took an approach where we were able to knock out or knock down just expression of the clock gene only in this area of the brain, the ventral tegmental area, the VTA. And we saw the same thing, which was an increase in dopamine cell firing. So this really suggests that it's clock in that region of the brain that's controlling them having too much dopamine, basically. We also found that this was time of day dependent. So when we did in vivo recordings and looked across different times of day, we found that they're actually they have the most change in their dopamine cell firing during the daytime and not as much during the nighttime. And what's interesting about this is that mice are nocturnal as opposed to humans who are diurnal. Mice are nocturnal. So mice like to be awake during the night and asleep during the day. And so, what this means is that the clock mutant mice are actually having these increases in dopamine specifically during the time when they're supposed to be asleep. They're not supposed to be out running around doing lots of things that mice do. They're supposed to be sleeping. And so, this is really an unusual time of day for them to have these surges of dopamine, which was really interesting. So we wanted to test this. We wanted to see, can we create a mouse that has this kind of manic-like phenotype just by stimulating dopamine cells only during the day? And so we used a technique called optogenetics, where we're able to activate the activity of these dopamine cells using a modified ion channel and a laser. So the laser light we put into the brain, we put it directly into the VTA. And when you increase the light, when you turn on the laser, that will stimulate the cell, and then the cell will fire, and you'll have that increase in activity. And we used a special kind of channel that has kind of prolonged activity, so we can activate over hours, basically. And so this is just showing the expression in the cells. I just like to show these pretty pictures of our dopamine cells and expression of this channel that's in the cells of the mice. So our paradigm was that we did surgery on the mice. We put in this ion channel and put in the fiber cable. We gave them then time to recover. Then we did 7 days of stimulation. So we did stimulation each day for 7 days of those dopamine neurons, either during the daytime or during the nighttime. And then we tested them in some behavioral assays and gave them some booster stimulations in between. So what we found is that if we looked at just their locomotor response to novelty, so we just put them into a novel environment and saw how much they like to run around, we saw an increase both with daytime stimulation and with nighttime stimulation, which you might imagine because you have increased dopamine that you've stimulated. But when we looked at things like anxiety or risk taking, so this is in behavioral apparatus called an elevated plus maze, where the mice have to choose between being kind of in a safe area, which is a closed arm of a maze, or in a scary kind of anxiety provoking arm, which is open and it's elevated, so they could fall off if they weren't careful. We found that the daytime stimulation led to kind of an increase in the open arm time and open arm entries, while the nighttime stimulation had no effect. And we saw the same thing in a couple of other assays. So when we looked at very similar kinds of tests where we're looking at the time they spend going into really brightly lit area or the middle of an open field, which is kind of scary for a mouse, that only that daytime stimulation led to increases in that novelty seeking or exploratory drive, or however you want to phrase it. versus the nighttime stimulation. So what this really tells us is that it's not about just having too much dopamine at any time of day that can cause these abnormal behaviors. It's about having too much dopamine at the wrong time of day. So having too much dopamine at a time of day when the mouse is normally supposed to be sleeping, it is not supposed to be out running around exploring novel things. And so we think this is really relevant for bipolar mania in the sense that, you know, it's often when your sleep is disrupted, when you're up at night, when you're doing things, you're, you know, having a rotating schedule or having or jet lag or something that's disrupted your schedule. And it might be that it's that surge of dopamine at the wrong time of day that's leading to this manic episode, as opposed to just too much dopamine all the time. We wanted to look a little bit more at what could be causing this. And so we did a whole bunch of different molecular assays, but to kind of just make it really brief, we found that clock binds directly to the tyrosine hydroxylase gene. So tyrosine hydroxylase is the rate limiting enzyme in dopamine synthesis. And we found that these mice have too much tyrosine hydroxylase, again, particularly during the day. And we looked at a little bit about how the protein is binding there. So it means that they probably have too much synthesis of dopamine because they have too much of the enzyme that is responsible for making dopamine. And then to test this, we used a compound that can inhibit tyrosine hydroxylase called AMPT. And I'm not going to go over all these behaviors, but essentially what we found is that if we inhibit that tyrosine hydroxylase, we can reduce their abnormal behaviors in some of those same assays that I talked about before, you know, the open field, the elevated plus maze. So this suggests that this increase in tyrosine hydroxylase is responsible for those abnormal behaviors. And then when we did the same thing at night, again, we had no effect, just the same as our optogenetic experiments, which again points to that's really that change in dopamine that's happening during that inactive period, the daytime for a mouse, as opposed to the nighttime. Okay, and then you know the other thing we can use the clock mutant mice for is to look at novel treatments or to figure out how existing treatments work because some of the treatments that we use currently for bipolar disorder, things like valproic acid or Depakote, as it's commonly known as, or lithium, we really don't know exactly how they work. And so we because they reverse the manic behavior of these mice, we can look at, you know, how are these medications working, and can we use that knowledge then to develop better medications with less side effects that are more targeted. So valproic acid, as I mentioned before, can reduce those sort of manic like phenotypes of the clock mutant mice. One of the things that valproic acid is known to do is inhibit these proteins called HDACs or histone deacetylases. These are enzymes that can affect gene expression. And so DNA is usually wound around these proteins into chromatin so that it can be really tightly packed inside the cell. And if you have HDAC inhibitors, it kind of loosens that DNA and allows for genes to be activated. If you have HDACs, it kind of closes it up, and then they're not able to be, those genes aren't able to be accessed. So VPA is known to be an HDAC inhibitor. And again, this is a ton of data on here. I'm not going to go through all of it, but I thought I would put this slide up from this paper if anybody's interested in really going through all the data. But basically, we went through a whole series of different HDAC inhibitors, and we found that that valproic acid seems to be working in the VTA, first of all. So it seems to be working in those dopamine cells, and it seems to be working through a particular HDAC called HDAC2. And that we know that because if we inhibit just HDAC2, we can also reverse a lot of those manic-like behaviors. We also found that if we overexpress HDAC2, it can prevent the actions of valproic acid. And so this really suggests that at least in the short term, what valproic acid is doing is it's inhibiting the activity of this protein called HDAC2, and then that's changing dopamine cell activity and it's reversing manic-like behavior. And so this leads us then to like HDAC2 being a good therapeutic target, although HDAC2 still affects a lot of different genes. And so a lot of our current work then is trying to nail down even more, like beyond HDAC2, then what genes does it influence? And then can we look at those specific genes and really try to figure out which is the one or two genes that's really necessary for this effect. The other way that we think that these drugs are working is through stabilization of circadian rhythms. And so these are studies in valproic acid and in lithium um, that show that if you put either one of these drugs onto cells that express a circadian reporter gene, so these are basically, it's just showing circadian rhythms of that cell. So these are cells in a dish. You can get an increase in kind of the strength or the amplitude of those rhythms, as you can see here. Both lithium and valproic acid do this, which means that what they're doing is they're really strengthening that internal circadian clock. We found in cell culture that this is even able to do this if you have the clock mutation. So even when you have something wrong with your clock, even when you have this mutation in the circadian clock that that causes these manic-like phenotype, valproic acid is still able to increase those rhythms. And so we don't know exactly how it's doing it, but it's really interesting that it's able to do it. And we even see this in the mice themselves. So not just in cells in a dish, as you can see here, but if you give it to the mice, if you feed it in their chow, so you put valproic acid into their chow, then you can see that normally if you measure the activity of mice, as I mentioned before, the clock mutant mice either have a long period, you can see their rhythms kind of get long, or they become just arrhythmic. But if you put it in them on valproic acid, it can really stabilize their rhythms. So we think that perhaps in the short term, that what some of these medications are doing is that they are inhibiting that change in dopamine. They're kind of normalizing that dopamine, getting it back to where it should be, so that it's not too much at the wrong time of day. And then in the longer term, these drugs may be preventing episodes by kind of acting like social rhythm therapy, by strengthening that circadian clock and really synchronizing the circadian clock. And then that kind of keeps those episodes from happening. And the good news too is that there's a lot of compounds now that are out there that can influence the circadian clock. And so this is a compound that was created by Pfizer that inhibits casein kinase 1, a protein that's involved in circadian rhythms. You can see that this drug does kind of the same thing. It increases circadian rhythm strength. And in mice that have a compromised circadian clock, it can kind of synchronize their rhythms, either mice that have a mutation here or mice in constant light. We tested this compound on the clock mutant mice years ago, back in 2012, and it also helped to normalize their manic-like behavior in a number of these different tests. And so, so this shows that if you give a compound like this that can synchronize rhythms, that it can also be therapeutic, potentially in these behavioral tests. And there's actually a lot of compounds now out there. We're testing several in our lab. We have collaborators that are testing some in their labs um, that we know will influence the circadian clock in particular ways. Um, and this is just an example of one called nobiletin, which is a flavonoid. It's kind of a naturally occurring compound, which can also help to strengthen circadian rhythms. And so I think this is really an exciting time in terms of therapeutic development, where we're able to take some of this knowledge that we have from existing medications, from our basic research experiments, and start to figure out, are there some additional treatments that we can develop that will target either those changes in dopamine or target the circadian clock, and potentially be therapeutic in the future. So to conclude what I've told you about, so circadian rhythm disruptions are strongly associated with bipolar disorder, and rhythm stabilization for a variety of methods can be therapeutic. We're starting to try to understand those seasonal changes in that can lead to bipolar disorder episodes. I'm really excited about this new work. I think this is really cool. And we found that yeah, there seem to be all these genes that are peaking in the summertime that are involved in mitochondrial function that might be related to changes that lead to mania. Um, there are a lot of seasonally rhythmic genes in major depression also, and they're more prominent in a different brain region in the anterior cingulate, and they seem to peak more in the fall and in the winter. So we're really interested in getting in and kind of teasing that apart. Um, we really love working with the clock mutant mice. They have this really strong phenotype that resembles bipolar mania. We found that it's largely caused by this increase in dopamine transmission at the wrong time of day. So having these surges again when the mice shouldn't really be out doing a lot of stuff that would cause surges in dopamine. This is leading to these abnormal behaviors. Lithium and valproic acid both may act acutely through reducing that dopamine signal. I didn't really show you the lithium data that we've published years ago, but they kind of influence dopamine. Lithium influences dopamine via a different mechanism, but similar kinds of results. And we do think that rhythm stabilization is kind of maybe a long-term way that these drugs are acting. So with that, I will stop. I want to make sure I leave time for questions, and thank the members of my lab who do the work, as well as our funding agencies, the NIH and some of our other foundations that we work with. We also really want to thank our collaborators and the people who, most importantly, the people who donate tissue to our brain bank, which is usually at a very difficult time. We're really grateful to be able to use this tissue for some of our studies. So thank you very much. And let me know if you have any questions. Should I stop sharing?
Yeah, go ahead.
Okay.
Yes, if anyone has questions, you can go ahead and ask them in the Q&A. We have about 15 minutes, so we can definitely do that. Thank you so much for sharing.
Mm-hmm.
Yeah. All right. Can you see these?
Oh yeah. Okay. So thank
Okay,
you for
perfect.
sharing. Would love to know the details of the lithium study. Yes. So as I showed in the beginning, we found that lithium decreased dopamine cell firing. And we did a number of gene expression studies and found that lithium seems to impact a gene that's co-expressed with dopamine, or a protein called colchicystokinin, or CCK. And basically the way CCK acts is it's released with dopamine, and it acts to go back onto the dopaminergic cells and kind of shut down dopamine levels basically. And so we found that lithium really seems to increase the levels of CCK a lot in the clock mutant mice. So we think that as opposed to this HDAC mechanism for valproic acid, we think lithium may be acting through CCK, but at the end of the day, they're kind of doing the same thing. They're both like shutting down that excess dopamine. But you can look at those papers if you're really interested. Would you advise using light therapy in the fall to mitigate depressive episodes? Light therapy, as I mentioned, can be really great for some people. It can be a little problematic. I would suggest definitely working with a physician to do light therapy. Some people have found with bipolar disorder that it's a little bit better to do light therapy kind of later in the day, like kind of mid morning or afternoon. But I would really, you might think about doing that, but I would really do it under the supervision of a physician just to make sure you don't have any unwanted side effects, especially that could trigger a manic episode. Has there been any credible study with natural supplements? Um, I'm not sure about natural supplements, but there is actually quite a bit of work now on the ketogenic diet and bipolar disorder. So we actually have a project in the lab on this right now. Um, it's sort of started as like a lot of anecdotal reports that people who were on a ketogenic diet, which is, you know, a diet that's low in carbohydrates and high in fat, um, that this diet was beneficial for mood. And now there's been some clinical studies, some clinical trials of people on this ketogenic diet. It's been used for about 100 years for the treatment of epilepsy. And a lot of the drugs that have been prescribed for bipolar disorder also happen to be anti-epileptic drugs. And so there's kind of this interesting correlation there. But you could look up some of those studies. They've been actually really promising for certain people. Being on this ketogenic diet may be therapeutic, but it's still really early days. You know, there's still a lot of work that needs to be done to figure out who responds best to this diet and how best to implement it and things like that. So again, something to consult with a physician about. Any studies with schizoaffective related to circadian rhythms? Yeah, so we have done a number of human postmortem studies on schizophrenia, and looking at differences in rhythms in gene expression. We haven't really done schizoaffective specifically, but some of the subjects that we have in our schizophrenia studies also have schizoaffective disorder, so it's a little hard to tease it apart. But we do find differences in circadian rhythms and gene expression in those same prefrontal cortical regions with schizophrenia, that are circadian differences. So not seasonal differences, but circadian differences that are really interesting. And also a lot of them are related to mitochondrial function. So it seems like people with schizophrenia may have some dip in a lot of um, their energy producing genes, and also in a lot of their genes related to neurotransmission, mostly during the night. So it's really interesting. But we haven't looked at schizoaffective specifically. Thank you for your talk. Do you have any more information? Those novel treatments being developed for pharmaceutical use. Yeah, so some of those treatments did start to go into clinical trials, but they're still really in progress. Um, so the pharmaceutical industry has sort of been, you know, their interest in bipolar disorder has sort of waxed and waned. They sometimes become interested in it and sometimes they don't. And so some of these drugs haven't gone very far. Like that Pfizer compound that I mentioned, they decided not to pursue it after it had some really good initial safety trials. But um, hopefully the pharmaceutical industry will become more interested in getting back into neuroscience in general and also into this space. But yeah, I don't have any specific knowledge of how far some of those treatments have gone into clinical trials at this point. Do regions in the US with low vitamin D levels increase the complexity of medication treatments? So I'm assuming you mean like regions in the northern climates where people get less sunlight. Um, there, yeah, there are some indications. Well, vitamin D is obviously like really, really important just in general for health. And a lot of people are found to be vitamin D deficient. And there are some indications that vitamin D does interact in ways with the circadian genes. And so, but I don't know of any particular studies that have looked at US regions specifically and compared them to other regions. But vitamin D obviously is very important, and sunlight is very important, you know, especially morning sunlight. So if you can get out, get some morning sunlight, you're going to help your circadian rhythms and help your vitamin D. So that's always a good thing to do. If interested, how does one go about donating their brain to your specific program after death? I live in the West Coast. So our particular yeah, so this is about donating your brain if you where depending on where you live. There are brain banks in a number of places across the country. Our particular brain bank only takes cases from Pittsburgh. So we go through our coroner's office here in the city of Pittsburgh, and that's where we obtain our brain samples. But the National Institute of Health has a brain bank where I believe that they collect from all over the place. And so I would contact the National Institute of Mental Health and ask them about their brain donation program. UC Irvine, I know, also has a brain bank, and so that might be a place to look as well. Any differences in clock genes in bipolar disorder 1 versus 2? That's another thing we haven't really been able to tease that apart in our brain bank. We unfortunately don't have a lot of people diagnosed with bipolar 2 in our brain bank. We do have, though, there's a very large study that's being funded by the BD Squared Foundation that has a very large postmortem component of it that is coming from some brains in New York City and in Pittsburgh and some other regions. And hopefully they'll be able to answer that question a little bit more because they'll have a better, yeah, they'll just have a better sample size than we do. We don't really have the sample size to be able to discern differences between bipolar 1 and bipolar 2, unfortunately. Um, I've been told that in the summer, 12 hours of darkness can help reduce mania. Do you subscribe to this as a method of controlling spring summer mania? Yes, there's a lot of people who are really interested in dark therapy for mania in particular. And there have been some really nice studies that have suggested that that really trying to get that extended dark period, particularly in the summer, can be really beneficial. Again, anytime you're starting any kind of new chronotherapy or anything, you want to consult a physician to talk about it. Um, but there have been some publications that have suggested that if you can get that dark therapy, that complete darkness in the evening, particularly in the summer, that can be really beneficial. I know it's very difficult depending on where you live. You might have light for really long periods of time. But there are some studies that suggest that can be beneficial. Are there any findings related to the impact of circadian rhythms on OCD symptoms and bipolar disorder? Not that I'm aware of. I don't know of any studies that have specifically looked at OCD symptoms and circadian rhythms. We do, yeah, our brain bank also is very limited in OCD subjects. And especially, yeah, OCD that's comorbid with bipolar disorder. So that's something we haven't been able to really look at. But it would be interesting to look at that and see if there was anything related to this. So I think that's all the questions. I don't think I see any more.
Yes, I think
Okay.
that's it. We can go ahead and wrap up for today, but I just wanted to again say thank you so much for sharing, for your time. Thank you everybody for joining us. And I did see one comment about the recording of this. This will be available on our website as it always is. So you can go there and search under, I think it's educational videos, and you can find a recording of this if you want to go back and listen again. But that's all for us. Thank you so much.